So if you are watching this video, that means you agreed to come on my little science stream in the future and do this, but with something you know, right? So the point of these videos is for me to get to talk to you, very intelligent people in STEM, and ask you about something that you feel very comfortable with, that is very common knowledge to you, that you probably think everybody knows, but in reality, that's not true, right? So it's a place for you to showcase your knowledge, to showcase your personality, and then possibly your art skills if you decide to be the artist in one of these amazing interviews.
I don't have art skills.
Rules are, I'm just going to talk, he's going to draw.
Yeah.
Okay, so DNA extractions. What are they? Let's find out. So, when you start any extraction, what do you need? Wait for you to write that out. The first thing you need is your sample, right? And that can really be anything. You guys have seen CSI Miami. They use blood. They use semen. They use hair. They use saliva. They use fingerprints. That won't have DNA, but you know what does have DNA in it? Dead skin cells, and that's what you can find in a fingerprint. You drew a mushroom. You know where you can also find DNA? Mushrooms. They have cells. Mushrooms have cells. They do. You could get mushroom DNA, and then you can identify what species of mushroom it is. It's really cool. That was totally correct.
That's a cell right there.
Okay, yeah. And so within your sample, you're going to have cells. You're going to have a numerous amount of cells. You're going to have a lot of other crap that you don't really need. Like, I don't know why I was going to say iron or debris, dirt, right? You're not interested in that. You're interested in the DNA. So what you're really looking for are these cells. Within these cells, you're going to have your nucleus, which our artist is going through it. So inside of your nucleus, right, if we're talking about a human cell, I guess is what we're doing, eukaryotic cells, which are a bit more complex than prokaryotic cells, right? You're going to have a double, oh, you're going before me. You're like, well, I want to...
You were talking about DNA?
Oh, this is great. So inside your nucleus, you're going to have your DNA, which is your classic double helix, which is what this amazing artist is going. And you're going to have your A, T, C's, and G's, your adenine, thymine, cytosine, guanine. If you want to get fancy, uracil, but that's something else. We're not going to get into that.
That's RNA.
Yeah, it's RNA, which is why I said I'm not going to get into it right now. But you have your A, T, C's, and G's, and those all hook up with each other to make a nucleotide sequence. And that is what makes up your DNA, which is what you were so interested in, because if you would make a circle for me, because these sequences make up a full genome. It's like a human genome project. If you like ever, I don't know, did 23andMe or whatever, and you send them your saliva, and then they are looking at the human genome, and then they can go, oh, cool, this section codes for you might lose your hair. This section codes for you might sweat a lot. You're short, you're tall, you're fast, you're slow. I'm going like red fish, blue fish, green fish, go fish. Dr. Seuss? Yeah, great. So you have your full genome, but what I do, because you can technically like do full metagenomics, which is your full genome, but what I'm looking at is a chunk of that. So now we're zooming back out. You have your sample. Nick, what sample are we going to choose?
I'm
Hobbs.
gonna erase.
What sample?
Erase that.
What do you want to choose for our choose your own adventure?
We're gonna go with toenail fungus.
No. Okay. So
Here's the nail, and it's on a toe.
Okay, you have athlete's foot in this scenario, and
That's not a nail fungus. There, this one's all.
Okay, what is it then, dude?
Oh, I have no idea.
Why is this what you chose?
You said choose something.
Okay, so you have a toenail fungus, and you are using different antifungals, but you can't. I don't know why this is the story, but I'm going with it. You cannot destroy it because the antifungals aren't working. Stop drawing feet. Just stop. This is not what I wanted to do.
Anyway.
So you're like, oh no, I can't destroy this fungus. I'm going to now have to ID it. So what you do is you go and you take a nail clipper and you a big chunk of that infected toenail. And you take that chunk and you put it in a sterile plastic microcentrifuge tube. You could use different sizes of plastic tubes, but because it's such a small piece of infected fungal toenail, you're going to put it in a small sterile tube. The difference between sterile and clean, because you need to know that sterile, it doesn't have any other DNA in it, right? It's DNA, DNase, RNase. Those are little things that go nom 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 and destroy DNA sequences or RNA sequences. There's none in there. Yes, great. So there's nothing to contaminate your sample, so you won't amplify that, which I'll talk about in another video, but you also won't destroy your DNA. So very cool. Great. So now you have your sample in there. You know what else you got in there? Yeah, okay, you have your fungus on the toenail. We're going to keep going back to this. This seems very personal to you. You
have to call me out.
You chose toenail fungus. You're going to add in mechanical and chemical weathering agents. So what is a chemical versus mechanical weathering agents? Let's dive in to that. Great. So chemical is exactly what it sounds like. You're going to be adding a detergent, which is your lysis buffer. Think about, like, I guess, soap, which is going to put its little hydrophobic tail into the cell membrane, because we were talking about how there's cell wall. Wait, nucleus is inside of the cell. You need to break into that cell so you can burst out the nucleus. I don't know, I'm fighting stuff with this. Have your lysis buffer. It's going to poke its little tail into the cell wall. Wick, you want to explain what that is right there?
Oh, okay. One second.
Because we're getting really into drawing.
This right here is a cell wall.
Uh-huh.
So those are like carbons attached to fat tails.
Okay.
And then they...
Oh, you erased it.
I erased it. So
that's okay.
I'm sorry, I have to draw my detergent bottle. So back to our fatty acid tails, which form our cellular membranes, which keep everything out of our cells. This right here, though, is the detergent. We'll say it's a chemical, and it pierces right through here.
Great. So now you have made little holes in the proverbial fence, skin fence. That is your cell wall.
Yes.
Great. So now you can get to the stuff inside, which is what you're really interested in. But to help that along, you're going to also do mechanical weathering. And to do that, you're literally going to put little pieces of rock, so like sand, gypsum, sand.
Oh, gypsum.
Yeah. Granite. I meant granite.
Not gypsum.
Not gypsum. Oh, shoot. Is it gypsum?
No.
It's a little purple bead. Little purple bead. Looks like a little rock. It's very pretty.
So it's sand.
Nope, it's not sand. The sand is white. Anyway, okay. It is a rock. I will edit it into the video. I'll go like, it's that.
Think of a giant cell.
It did it. It's there, right? It's right there.
Then
you can't see it, but you will post.
Purple.
In post. I know what it is. I do this all the time. Yeah. But anyway, yeah. So you add it into your sample along with your chemical lysis buffer. And you know what else you add? Some salt buffer. And that keeps the pH real comfortable. So the DNA, when it goes like, oh my God, I'm free, it doesn't just die and disintegrate or break up. So you got it stable. You got that lysis buffer. You got the mechanical little pieces of rock. And then what you're going to do, you're going to seal it real good. You're going to put it into, so I've explained it before, but a boba machine. So if you've ever had boba, they have these little like robots and they grab the top and bottom of the drink and they go like, ah! And that's what you're going to do to your sample. But like a whole bunch at once. And it really does look like that. It's a boba machine. It just it's like, I swear that's how, I don't know what came first, but I really think that
boba didn't come first, people. I'll let you know that right now.
I think boba then saw someone do a DNA extraction and went, you know what? Wow. That would be so good for DNA. Anyway, I really think this is true. Like a conspiracy, maybe. I don't know. So you've lysed the cell, and now your nucleotides are like, I'm in solution. No! But they're also really dirty. They're really dirty. There's a lot in there. There's all of these parts of the cell that we didn't mention because I didn't want to, because I don't remember every part of the cell. And it is there.
Yeah, your broken cell.
Cool.
You got your DNA.
Nice. What else you got in there? Oh, you know what you'd have? Toenail.
You do. You have pieces of the toenail still.
And that's sunk straight to the bottom.
Yeah.
Right?
Yeah.
Because again, like, and these are really tiny tubes.
Oh,
you put the sample in the centrifuge machine. A centrifuge machine uses centripetal, not centrifugal, right? Centripetal force to then make all the heavy stuff fall down. You can think of it like, what did I use? Oh, okay. Astronauts, when they go into space, they have to learn how to handle all that centripetal force up in space. So they put them in a machine that goes like, wow, G-force, thank you. It's like that. Heavy things go down, light things go up. That's how you separate it. So now you have all of your debris in the bottom of your sample, which is great because you have your DNA in solution. So what you're going to do is you're going to aspirate your supernatant. You're going to take out the liquid part, which is the supernatant, and put it into another sterile tube. A lot of tubes. Go from one tube to the next tube with your pipette, which is your little handheld vacuum, which we love so much. You can turn it. That doesn't make sense. You can turn the top of it and go, oh, I want this much. I want more in there. I want less. It's amazing. You've got all these little tips you can put on there. It's like so satisfying, especially if you have like a little bit of OCD like I do, because you can organize them in lines. Ugh, it's so nice. So now you have your DNA in solution in your sterile tube. And it's free flown. It's having a great time. But you still have other crap in there, and you really just want the DNA, right? Because you still have all of that other solution in there, probably like the buffer, etc., right? So what you're going to do is you're going to add more salts, and your salts are going to bind to the DNA, and they're going to go to the water.
What kind of salts?
I don't remember. Did I say it? Did I remember? Did I say what it was before? You're gonna put salts in there, baby.
Yeah.
Tris HEL is the buffer. I don't remember the salts. We can put a question mark and I'll do this again. This is the salt you put in. It's this one.
And
Great.
then it makes the DNA.
And it makes the DNA
solid.
just fall. So now you add it in. It looks milky at this point because there's a lot of stuff going. There's a lot of stuff going on in there. They're grabbing each other. I see what I'm saying now. I heard it.
Okay.
It's okay. You have your milk. You have your stuff.
You have the precipitated
You have the
DNA.
precipitated DNA. So then what you're going to do is you're going to put that in a centrifuge. So you're going to put it in a centrifuge. Put that in a centrifuge. This is where you could like do the little lasso thing and then pull the image down.
Oh, no. We're drawing everything
You're gonna redraw
from
it.
scratch.
It's going to take longer, but I appreciate the effort that you're putting in.
That's effort right there, baby.
Good. So, great. You're centrifuging it again, and what does that do? It pulls all the heavy stuff to the bottom, and all the light stuff that's liquid stays on top. Amazing. So now at this point, you hopefully have a pellet, and that is your DNA. That is exactly what you want. It's your A, T, C's, and G's, and hopefully it's not super chopped up. It's good quality, blah, blah, blah. So at this point, you go, oh, wow, I'm done. You're not done. You still have 3 more steps. But, lucky for you, whoa, they're pretty easy.
So you gotta extract more of the supernatant off?
Oops. Yes. So at this step, you have your pellet. Hopefully it's clear or milky white. And you're going to take out... Why do you keep smiling at milky white, dude? You're going to take out the liquid. So you're aspirating the supernatant, and now you just have the pellet. But you still need to clean the pellet because you might have the lysis buffer on there. You might have some salts. You might have all this other crap that you've put in there still on your pellet. So you're just going to add enol. An isopropanol, an ethanol, and then it's going to grab on to anything else that is in there and it's going to bring it into a liquid solution, and then you're going to, no, you're not going to, yes, you do centrifuge it again. You're going to centrifuge again. Okay. So you're going to centrifuge it again.
It's like a bomb.
And then you're going to aspirate that out, and you're going to do it for a second time, usually with 70%. First time is with 70% ethanol.
Times 2.
Second time is 100% ethanol, or again, you can use isopropanol, depends on what you're doing. And there's like ratios that I really am not getting into.
Don't worry about that.
But if you're interested, ask me. I'm here for you. So, where am I at? Okay, great. So now you did your last centrifugation step with your ethanol, cleaning that sample, and you're going to aspirate that out. That's my noise. So good. You took out that ethanol, and now what do? You let that DNA pellet dry. So this is what it does. This is what I think it does when it's sitting there. I do this too often. Thought about this a lot. But yeah, so now it's just drying. And you want it to get to a point where it's like supple. You know, you don't want it too dry, because if it's too dry, it cracks. And if it cracks, you can damage the DNA, or it won't go back into solution when you add the last step, which is the elution buffer. And your elution buffer is going to let that DNA go back from a solid phase back into an aqueous phase. So it's now partying, hanging out with the water or whatever you're using. You can technically use very sterile water, or you could use a buffered solution, which would be like a tris, which is, again, just going to make the pH really, really comfortable for everything. And now at this point, you have your extracted DNA, and it's beautiful, and it's clean, and you're going to nanodrop it. We're going to do nanodrop really fast, because actually I think we should have added this. Okay. So now you have your DNA, and it's clean, and you're like, oh my God, I did it. I'm going to go home. I'm so good at my job. And then you remember that you maybe did a really bad job, or that the sample you took had no DNA in it at all, and you did a really fantastic job, but the world does not smile on you today. So what you must do is Google it. No, what you must do is nanodrop or qubit your samples. And this is just reading the amount of DNA you have with fluorescence. So you have this machine. Do you know what it looks like?
No.
I'm going to explain it. We're going to see how similar it is to the real life one. It's like a rectangle, but then you have an iron on it. So you have like a circle and an iron on top of the circle, and then you've got a rectangle, and then you've got a TV. Yeah, you know what? It's pretty good. Pretty good. What you're doing right now? Pretty good. Just a TV on top now.
Like
a backboard of a basketball hoop on the top. On the top of it, like a basketball hoop backboard, like a backboard. Yes. Holy crap, you made a nanodrop. That's what it is. And what you're going to do, first of all, is take your elution buffer. No, literally, that's what it looks like. You're going to take your elution buffer, which is your pH buffered solution or your very, very sterile water, and you're going to grab like 1 microliter. I am using liters. That is how I do my stuff. So
that's how the world should do it.
Don't at me. I hear people say that. Anyway, you take a little piece of it, you put it on the little nubbin. So there's a little nipple, Nick.
Where?
Underneath the iron. So
Oh,
you
I have
lift
to
up
lift
the
that
iron.
Okay, let me draw my
You can just like
masterpiece.
pretend that there's a lifted iron there.
No. I'm drawing this thing again.
Wow, it's so good.
Oh, I gotta draw because.
Why don't you just say there's a lift?
No. Shh. It's okay.
I'll edit it. I'll edit this down.
Don't edit it down.
I'm gonna have
Nope.
to edit.
No. Don't edit it down. It's gonna be perfect. So here's our little iron friend. You don't have to do that. Here, oops, it's a little too big. I lifted it though.
Yes, and so when you lift it,
And
there's a
then-
little nipple.
This nipple
Beautiful.
is a big nipple.
That is what it looks like though. Okay, great. So now you're gonna put your elution buffer, which is a very small amount. A microliter is like the tip of a pin, if you were to look at it.
Yeah.
Yeah, if the tip of a pin was liquid, that's what it would be. And you're going to put it on the tip of the nipple. And then you're going to close the iron. And then once you close the iron, oh my God, phosphorescence is going to shoot, like light is going to shoot in there. There's a spectrum going on. There's some physics going on that I do not understand. Spectrum shenanigans. And that is going to quantify the amount of DNA in there, which should be 0 because it's your blank. And after that, you are going to lift the iron. You're going to clean off the nipple. And then you're going to add every single sample alone. You're going to do it to all of them. So if you're in a lab and it doesn't have a lot of money, no shade, but you're going to only have one nipple, and you're going to have sometimes like 8 plates of samples, and each plate has 96 samples. So then you're going to nipple count every single sample, and it takes so long. And if you have less than 1 nanogram, which is a small ass amount, if you have less than 1 nanogram of DNA, you got to redo those samples. And that's it. And there's also quality, so you got to see, like, if you're looking a little bit concerned right now, Nick.
That's crazy.
Yeah, I mean, that's how it goes, though, right? So there are fancy versions of this, which I have not gotten to use because I am in academia and we pour out here. But I've heard from other people who work in like medical facilities, who've worked in industry, there are these plate readers. Ugh. You just put a plate in there and it goes like, bah, 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 bah. And that's it. And then it tells you it has so many nipples. And it just tells, I don't
like
actually,
a cow.
no, I don't think that's how it works for those, because they're single samples.
Let's just assume.
It's not right. Anyway, there's a way where you can do plate readers, but the way I do it are single tips, which is, it's time consuming, but there are way more efficient ways to do it. But this is a good example for you to understand how to do it with a single. Yeah, and that's really it. There's like 8, there's then like lights, making you sound like it's really Christmas tree themed. Core, is that what kids say? It's coded. It's not. It's a screen. And then it goes, oh, you have 1.8, which is great because that means you don't have a lot of crap in your DNA. And there's another category, and if it says a certain number, that's good. If it doesn't say that number, really bad. And then you got to clean your DNA, and I'll get into that in the next video because that's all a part of the second step, which is amplifying said DNA. You're looking at me.
PCR.
Polymerase chain reaction, which again, if I were to explain that right now,
it'd be a lot.
It would take so long, and it would make me very tired. But that's it. That's how you get DNA. So now, do you want to like draw a little DNA, a little happy DNA? A little happy DNA. Like 5 nanograms of that stuff. So you got a good amount of it from your toenail.
It wasn't my toenail.
From someone's toenail. And now we're going to know what it is. And we're going to save that person's foot because their foot was going to fall off. Because that fungus was going to spread and they were going to die. And now they won't. I have a rash. You're going to see this. That's what that is. It's poison oak rash. Side note, don't be scared. Rashes are normal. It's common. It's beautiful.